A 39-year-old man lay brain-dead in a Chinese hospital. What happened next shocked the medical world. Surgeons replaced his left lung with one from a genetically modified pig. For 9 days, that pig lung kept working inside a human chest. This wasn't just another medical experiment. This was the moment everything changed. Here's why this matters. Every year, 173,000 organ transplants happen globally. But the need is massive. In Europe alone, nearly 4,000 people are waiting for lungs right now. 216 of them will die this year just waiting. But lungs are the ultimate challenge. Unlike hearts or kidneys that work in closed systems, lungs are constantly exposed to air, bacteria, and the outside world. They have this massive surface area packed with immune cells that attack anything foreign. That's why lung transplants have the worst success rates of any organ. So when doctors talk about xenotransplantation, putting animal organs into humans, the lung represents the final boss. If you can make a pig lung work in a human, you've cracked the code that could save hundreds of thousands of lives. The team at GUANGZHOU Medical University didn't just grab any pig. They used a Bama Xiang pig that had been genetically engineered with 6 precise modifications using CRISPR technology. 3 pig genes were knocked out: GGTA1, CMAH, and B4GALNT2. These genes create molecules that human immune systems immediately recognize as foreign and attack with everything they've got. Think of them as giant not-human signs painted on every cell. Then they added 3 human genes: CD55, CD46, and THBD. These act like molecular diplomats, telling the human immune system, hey, we're actually compatible, and helping control blood clotting and immune responses. This 6GE combination represents years of research to create the most human-compatible pig organ ever developed. But genetic engineering was just the beginning. The patient was already brain-dead from a brain hemorrhage, with family consent for this groundbreaking research. The surgical team replaced his left lung while keeping the right human lung intact. Then came the most intensive monitoring in transplant history. Hour by hour, they tracked everything: oxygen levels, immune responses, inflammation markers, X-rays showing lung function. The immunosuppression protocol was unlike anything used before: 8 different drugs, including some experimental treatments. Here's what happened: no hyperacute rejection. That's the immediate, violent immune response that destroys pig organs within minutes in an unmodified system. For the first time in history, a pig lung was accepted by human immune defenses. But at 24 hours, complications began. Severe swelling developed, likely from ischemia-reperfusion injury, the same problem that affects human lung transplants. Days 3 and 6 brought episodes of antibody-mediated rejection, where the immune system launched targeted attacks. Yet incredibly, the lung recovered each time and continued functioning until day 9, when the family requested the experiment be ended. 9 days might not sound like much, but this represents a monumental breakthrough. Previous attempts at pig-to-human lung transplants failed within hours or minutes. This proved that with the right genetic modifications and immunosuppression, pig lungs can sustain human life. But let's be clear about what this doesn't mean. This wasn't a cure. The patient was already deceased. The immunosuppression was so intense it would be toxic to a living person, and 9 days is nowhere near long-term survival. The inflammation and rejection episodes show that major hurdles remain. The genetic modifications need refinement. The immunosuppression protocols need to become less toxic. And the preservation methods need improvement to prevent that early swelling. This lung transplant is part of a broader xenotransplantation revolution. Pig-to-human kidney transplants are already in early clinical trials with living patients. Heart transplants have been attempted in compassionate use cases. But lungs were always considered the final frontier because of their complexity and immune exposure. Now that barrier has been crossed. Companies like eGenesis are raising hundreds of millions to commercialize these technologies. The competition is heating up as everyone realizes we're approaching a tipping point where animal organs could become routine medical treatments. So when will pig lungs save actual patients? Experts estimate we're still 5 to 7 years away from trials in living humans for lungs, though kidneys and hearts are moving faster. The next steps are crucial. Optimize the genetic modifications, develop less toxic immunosuppression, improve organ preservation, and establish long-term safety protocols, including monitoring for pig viruses that could theoretically jump to humans. But make no mistake, this 9-day experiment in China just proved that the biological barriers can be overcome. The organ shortage crisis that kills thousands every year finally has a solution on the horizon. A pig lung working in a human chest for 9 days. It sounds like science fiction, but it's the reality that just changed medicine forever. The question isn't whether this technology will save lives. It's how quickly we can make it safe enough for the patients who are dying while they wait. The future of organ transplantation isn't human donors. It's walking around on farms right now, being genetically engineered to save human lives.